Hello, everyone. I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is Whole Genome Sequencing for Healthy Individuals, providing genomics-based guidelines for the optimization of care and lifestyle. And our sponsor is DNAnexus. Our panelist for today is Dr. Wayne Delport, Vice President of Technology and Bioinformatics for Human Longevity, Inc. You can type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar screen. And if you look to the tray at the bottom of your window, you'll see a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Dr. Delport. Please go ahead.
Thank you, Christy. Yes, thank you for this opportunity as well to be able to present the work that we've been doing at Human Longevity in the genomic space over the last several years, and really the focus here has been, how do we create a whole genome product, that can benefit people that are coming in for a healthy screen, basically. And so before I can get into the details of that, I think I should be providing some details on what Human Longevity is about. And really, Human Longevity, if you don't know, the company was founded several years ago on the premise that the genome was going to revolutionize the practice of medicine and make a significant impact on integration of genomics in medicine. I think we've seen a lot of that in the oncology space. We're not entirely convinced that we've seen that in the primary care space. And so really, Human Longevity was established to try and address this problem. And part of addressing that problem is creating an internal clinic called the Health Nucleus. And so really what the Health Nucleus is doing here is they're trying to change the way medicine is practiced. So if you compare on the left-hand side of the slide here, we have your sort of conventional healthcare model to the sort of next generational healthcare model that's very dependent on data-driven insights. So in the conventional model, the code part of it comes from family history or interview with a client or with a patient, whereas in the next generation part, this is coming from a whole genome sequence, or from where your risk is established from analyzing your genome. On the conventional healthcare model, the core exam consists of a physical exam, and typically the exam is only does things that are related directly to your symptoms. So if you're going for a blood test, your physician draws or does the blood tests that are appropriate for your condition or for your symptoms. Whereas on the next generation side, it's about deep phenotyping and deep phenotyping across multiple modalities, and this would include imaging, whole body MR, in some cases where appropriate CT scan, and other biomarkers as well. So extraction of a full panel of blood biomarkers, and at multiple time points. Every time you come back for a visit, another full panel gets done, and even metabolomics, and in some cases even microbiome panels as well. And so the point of this is that previously conventional model, a physician would integrate this information manually, whereas under this Health Nucleus practice, a physician integrates these data modalities, but is empowered by artificial intelligence. And so this is very much the focus of what HLI is working on at the moment, is enabling this physician interpretation that's driven by analyzing deep data from both genome and from other modalities. Today's talk, we're focusing mostly on this part of things, on the whole genome sequencing part of things, and how do we extract value from a whole genome and put that in front of a physician so that physician can then execute client care in an informed way. Just to highlight some of the actionable results that have come out of the clinic thus far, right? So this is based on a paper that was published in 2020, which describes this approach to medicine. And really, I'm just going to highlight a few, not going very much details on them. Nearly 7% with severe cardiovascular risk. Nearly 30% with elevated liver fat, and of course these people are all at risk of developing type 2 diabetes, and then 24% with a rare genetic mutation that can directly impact their health. And then, of course, a large percentage that are genetic carriers for disease. So this is informative for those clients, but also informative for the children and the families of those clients. And so today we'll be focusing mostly on this part of the year. So this this development of a healthy genome product, when we started looking at this, we looked at what was available in the market at the time, and really there was nothing that met the requirements of what we wanted to be delivering to clients. And so we took on the approach of developing this internally. Our requirements here is we needed this to be engaging for both clients and for physicians. The language needed to be written in such a way that a client could pick up their genome report and understand the relevance of what they were looking at. But at the same time, the physician needed to be able to pick it up and feel that there was credibility associated with what we were putting in front of them. Of course, we needed sections on monogenic disease. We needed a prognostic risk assessment. We needed a section on pharmacogenomics. We wanted this to be a whole genome scope. And we also wanted to include some recreational content in this as well. The other requirements we've set here is we needed this to be cut clear, right? We operate as an LDT, which means the physician has to place this order, and a cut clear laboratory issues the final report. And then finally, we didn't want this to be a diagnostic, so we didn't want to be able to deliver this with a phenotype in mind. A lot of the genetic testing space out there is very diagnostic driven, and we wanted to make this more of a prognostic, health and lifestyle optimization, and also an assessment of current state of risk. And so we've been through various iterations on this product over the last 5 years, and this is where we are right now. Is we have something that runs about 20 pages long, has 4 main sections on hereditary disease risk, pharmacogenomics, a health predisposition section, and nutrition and wellness traits. And this is being issued by an entity that we run inside of Human Longevity called Human Longevity Clinical Labs, and that's a CAP clear. We have our CAP and clear licenses for that entity. Before we can really go into the development of each one of those components of the products, I want to talk through a little bit about the sequencing assay. Prior to 2019, we were doing our sequencing in-house. We had some changes in the company that resulted in us getting rid of a lot of the instruments and changes in direction. And so what we did here is we partnered with a CAP-clear validated reference laboratory that does our whole genome sequencing for us. And so what they're doing for us is 30x mean coverage, using a PCR-free preparation, library preparation. They also run a QC array for us, and this is for the intent of making sure that there's provenance of the sample within the lab. And their deliverable to us is a FOSS-Q that they deliver to our clinical lab. And everything downstream of that, the analysis and reporting, is being done inside our clinical lab. And so we've done the necessary validations here. We've done the necessary validation on the whole genome sequencing that's coming from the reference lab and internally on our pipelines. And so we can assess SNVs, small insertions and deletions. The sensitivity and specificity numbers here wouldn't be surprising to anyone that's running one of the modern combination aligners and variant callers. Insertions and deletions, yes, this is a little bit low. I'll talk that through on the next slide. So in terms of how we're doing the secondary analysis part, and really we still consider this as very much a commoditized part of the workflow. Sequencing part and alignment and variant calling is really commoditized at this point in time. And so currently we're running Illumina's HiSeq, an earlier version of the HAS software suite that include Isaac Liner and Starling. This was really selected at that point in time for speed and for cost. When we did that initial evaluation, the equivalent GATK pipeline was running at an order of magnitude more expensive, an order of magnitude more in time. And because we were building this in cloud, we were paying for that. In future, right, what we're moving on to, and this is some of the focus of this talk, is we've moved over our platform onto DNAnexus. And Sentient has partnered with DNAnexus and has provided the secondary analysis tools there. They're very easy to just pick up and start using directly. And we've picked up one of those, done a few tweaks on the parameters in there, and that's what we will be using going forward. We have a validation study in process on this, so we're not completely switched over onto this platform yet, but we will be in Q4. For both of these, we're computing about 150 QC metrics, of which we track 19 actively, and there's 4 strict pass-fail metrics. So your typical ones, we track this, contamination metric, and YX coverage is just another measure of QC that we can confirm against client gender to make sure there weren't any issues with the samples and in delivery or in the lab. So that really covers the sort of more commoditized base, and so I want to go through each of those sections that are represented or that I showed earlier. First, the hereditary disease section. And so for hereditary disease or monogenic findings, what we're reporting here is pathogenic and likely pathogenic variants and risk factors. Pathogen LPLPs, typically have high or moderate genetic risk, very rare allele frequencies, less than 1%. There's very strong support in clinical evidence, whether it's co-segregation and functional impacts of those variants. And the odds ratios typically run very high, like typically greater than 5. On the more risk factor side of things, there's sort of low genetic risk for developing disease. Typically, there needs to be multiple variants that co-occur in order for disease to occur. These are typically common with allele frequencies running greater than 1%. There is support by clinical evidence, and odds ratios run in the sort of range of 2 to 5, as opposed to greater than 5 for PLPs. And so how we define this is for every single release that we do, we have a predefined gene variant list. And so I'll talk through how we assemble that. But this variant list will remain in place until the next release. So any client that gets a version of the report will be getting a specific analysis against a specific variant release. And we strive to do at least one release per year. And in some cases, we manage to get more out. To talk you through how we produce or how we create this variant list, of course, you can't do manual curation on all of this, and our primary goal on this was to be able to do this, to scale this, and scale this using infrastructure and not necessarily using a lot of people. And so we built out a process here using that could do variant classification. Several other companies have done sort of similar things in this space. And so really the input starting on the top left here is any variant input. And so that could be a VCF for a client, or it could be an aggregation of several databases. And so the first question, moving from the left to the right across this slide, the first question we ask is, have we seen this variant before, and do we know what it should be classified as? Right? And this allows us to split it between, should it be a VUS, a benign or a likely benign variant, and in which case it's not reportable, going along the top, or should it be a pathogenic, likely pathogenic variant, in which case it is reportable, going along the bottom. If we haven't seen it before, the next thing we look at is we evaluate moving across, we evaluate population databases, and we ask the question is this variant high enough in any of the populations that are represented in that database for it to be considered a benign variant? And if that's the case, it will push into the non-reportable content. If that is not the case, it moves to the next section where we have a look at external databases like ClinVar and HGMD. And really what we have there is a set of trusted and expert sources that we've identified. And the question we ask is, do these expert sources all agree on the classification of this variant? And that also allows us to split it up between VUS, benign, likely benign, or pathogenic, likely pathogenic. Importantly, this often occurs that there's disagreement in ClinVar, and this is where we push it along this path of potentially pathogenic, and it goes for manual review and classification, which then feeds back into our variant database. And then finally, the last step, if none of those variants have popped out, if the variants as input haven't popped out from any one of those prior 3 steps, we go through our own variant classification module, where we've got an automated ACMG criteria in order to make a decision on whether it's VUS, benign or likely benign, or PLP, or potentially pathogenic. And so this is really how we run this for every release. And our input into this was nearly 1 million variants aggregated from multiple sources. And those sources include ClinVar and HGMD. It includes our own client database, variants we've observed in clients. And really, when we run this algorithm, this pie chart on the right here shows the breakdown along all these paths. So about 67% on one of the runs here went into VUS, LB, or B, right? 24% into PLPs, and 9% into manual review. The result of that is we get a reporting scope out, so I get about 1 million variants in, about 108,000 variants out. Those are all the PLPs and risk factors, and this just shows the breakdown of all the diseases that we're covering. So the majority are in hereditary cancer, followed by syndromic disorders, neurological disorders, going all the way to kidney disorders, and then some with much lower frequencies. How do we report this to clients? So this is just a snapshot view of our reporting. We have a findings section where you basically some general statistics. We report out to a client the number of diseases, risk variants that they have. This would be a monogenic finding, a medically significant finding. For example, like an autosomal, heterozygous autosomal dominant variant, and then we also report out the carriers. We list out the tables, the variant information in tables. We provide a variant interpretation, which describes the details and links that content to published examples or published evidence. And then, of course, we also provide some information about the gene. In terms of our findings on this, so if we look at the number of clients that have had a finding, and this is across a few thousand reports that we've issued over the last several years, about 12% have no findings at all, depending on how you structure medically significant. Anything between 14 and 38% of clients have medically significant finding. And so this would be, for example, like a heterozygous autosomal dominant or homozygous autosomal recessive. And so the difference between these two pies on the left and the right is that on the right, we're excluding some of the more common risk factors in making that assessment. But these numbers are an agreement. We see similar numbers from labs like coming out of Invitae. We've seen similar numbers from some of the healthy testing done at Mayo Clinic as well. For our pharmacogenomics section, we curate pharmacogenomics content. We of course, pharmacogenomics, we have star alleles, right? Star alleles are haplotypes. So building this on a SNV and indel validation is not the only work we have to do. We have to define what those haplotypes are. And so we get this information from CPIC and PharmVar. Importantly, we exclude any alleles from our reporting scope where there's ambiguity on the functional effects of those alleles. For our drug responses, we consult a number of sources listed here. And really, when we're asking something about a drug response, we're looking at sort of 3 levels of evidence. Like the first sort of lower level, so is there any evidence that this genotype has an effect on the drug? Like, can it influence plasma concentrations? And then 1 level up, is there an associated risk, right? Does this result in an associated risk? And then finally, at the sort of highest level, is there a specific recommendation? Is this an FDA drug label dosage recommendation? And so we collect that information and provide that to clients. Of course, for pharmacogenomics, we need to do a validation. And so this is where the CDC and in particular the GetRM program has been phenomenal in producing or keeping test sets updated. And the point there is to really provide a reference set of cell lines for pharmacogenomics testing. And so we've selected 30 of those cell lines that were representative of our allele scope. And we've done whole genome sequencing on them, and we keep that as our test set. Every time we do a new release, we'll run that as an additional validation. In addition to this, we also do a manually derived truth sample. So we actually, the genomic scientists go into a GBCF and they will manually call star alleles by observing heterozygous and homozygous variants that are in the scope of the allele definitions that we're using. For our reporting in pharmacogenomics, we're currently reporting for 9 genes, covering 246 variants, 63 drugs. Importantly, we report on copy number for CYP2D6. And so the CYP2D6 is responsible for 25% of prescribed drugs, like a lot of these include antidepressants and opioids, amongst others. And so that's why I think it's really important that we do copy number. For how we report this out, we basically, on the right-hand side here, we report out what is a therapeutic group. group, what is the gene, what is the inferred genotype. In some cases, we can do allele-specific amplifications as well. And then what's the inferred phenotype and drug, and what the recommendations are. Some of our findings from pharmacogenomics, I'm just pulling out 2 genes here as examples. For CYP2D6 on the left-hand side here, we see that if we're just looking at the phenotypes, about 76% of our clients that we've issued reports for are normal metabolizers. I must note that very recently, last year, the guidelines changed on this, and so now there's some additional phenotype categories that have been defined in the guidelines, and we're currently in the process of incorporating them into our next release. And those changes would actually result in the number of normal metabolizers going from 76 to about 50%. But this is consistent with the allele frequencies or phenotype frequencies that we see on CPIC as well. For copy number on CYP2D6, about 79% are normal, 17% amplifications. About 3% have a heterozygous deletion, and we've only seen 1 homozygous deletion in our clients so far. On the right-hand side here, SLUCCO1B1, just looking at the top pie of there, this is again a breakdown of function of alleles, right? And so about 50% of our clients have a normal function. Of course, SLUCC1B1 is metabolized as simvastatin, right? And so if you have any decrease in poor function, there's a potential for myopathy. And so this is where approximately 22% of our clients should be consulted on whether they're taking a statin or not. There's this rather big pie, the yellow pie there of 26% of unable to determine. I want to unpack that a little bit and explain why. I mentioned at the beginning that of the pharmacogenomics section that we weren't reporting out star alleles where the phenotype was ambiguous or the functional effect was ambiguous. And this is the biggest culprit here, is star 14 has a potentially reduced function. And so because we're not reporting out those phenotypes, it becomes unable to determine. It makes a big contribution to this unable to determine. We do still report out the genotype for those clients. We're just not making an explicit call on what their phenotype might be. The other interesting one is this one here, this combination of star alleles here. And this is cases where we couldn't unambiguously resolve the variance as a combination of 2 haplotypes. And in this case here, the variant that occurs in 1B and the variant that occurs in 5, they both occur in 15. So there's, you either have a combination, they're either in cis or named trans. And so in these cases, initially we were reporting these as unable to determine for the phenotype, but the phenotype for these is the same, and both of them would have a decreased function. And so now we're reporting out the ambiguity and we're reporting out decreased function because both possible combinations occur. Moving on to our health predisposition section, and really this is the polygenic risk section. Probably don't need to remind this audience, but this is really about looking at the heritability of common diseases, right? And if you consider this table here on the left, if we look, for example, what is the heritability and how much of that is explainable based on monogenic variants? And we know that in some cases, only like 5 to 10% of what we're seeing in heritability of disease is explainable by monogenic variants. And that could be one of 2 things. It could be that we just don't know all the variants that influence disease. If they are monogenic, we just haven't observed them. Or alternatively, variants work together. The disease risk is influenced by multiple variants of small effects, and that's what's being represented here on the right. When you have variants of very strong effects and they're rare, this is what we're reporting out in our monogenic section. But you can have lots of variants of very small effects that contribute, that are common and contribute to development of complex disease. And this is the polygenic risk section, what the polygenic risk section covers. So of course, when you're starting a product development for polygenic risk, right, we need to go through some study curation, and we use the combination of a data-driven and a curation approach here. Data-driven in the sense that we parse GWAS catalog to identify studies, and then the curation on those studies. And so studies have been published, curated studies are published in respective journals, typically by an established group with a track record in either in that disease or a track record in GWAS in general. We looked at studies where the phenotypes are well-defined. We looked at studies where the discovery cohorts were well-defined in gender, ethnicity, and age. We considered recently published studies and where the number of cases in the discovery cohort was sizable. And then also preferentially selected studies that validated in other cohorts. For our model curation, the way we approach this is, again, starting on GWAS catalog. We have all the effect sizes and all the effect alleles. We did some curation on that to confirm the variant count, confirm the variant details, confirm the effect sizes and effect alleles before implementing it in our product. We went one step further on this, and we did a validation of the work that we collected. So we had about 30 odd polygenic risk models that we curated and curated the content. And we went to UK Biobank, and we started with UK Biobank data and filtered 2 participants of European descent. We did case control assignment based on ICD-10 codes and health questionnaires for each disease, proceeded with calculating PRS scores from the genomic data that we had from UK Biobank, and then computed AUCs, odds ratios, and prevalence. And so these results are all summarized in a white paper that's shown there below. And that is linked in our report that clients, if they wanted to go and investigate this in any more detail, they would have the opportunity to do so. And so on the right-hand side, not surprisingly, diseases with very high heritability performed the best, like AUCs running like 80 plus. Some showed intermediate performance, and the list is shown over there, and then some were pretty low. And so we don't report anything where an AUC is less than 0.55. From a machine learning point of view, that's quite a low threshold cutoff. But remember here that heritability influences the performance of these models. And if there's a very strong lifestyle component, then these models won't perform very well. And notably, we didn't include any other covariates. We just include genetics into the calculation of these AUCs. And unpacking the validation a little bit more, right? This is just a snapshot of one example here. Of course, we can calculate a distribution of polygenic risk in our validation cohort. We can pull percentiles from that for any particular client here. Are you at the 80th or the 90th percentile? But we go a little further than that in the sense that we want to know what the relationship is between odds and a PRS percentile. And so to do that, we basically assume that those people with an average genome, right, in the 40th to 60th percentile are representative of the average case. And so anyone that has a lower percentile would have a less than 1 odds, and anyone with a greater percentile would have greater than 1 odds. We do the same thing for prevalence. We calculate like prevalence for each percentile. And so really the thinking behind this is that a physician can consult these sorts of curves, and if a client comes in and has an 80th percentile, they can talk them through, okay, so you're at the 80th percentile for this, but even samples or even UK Biobank participants that were at the 80th percentile, the prevalence of disease in those participants is only 8%. And so that's the sort of thing that a physician would engage the client in the delivery. The really strong point I want to drive on this is that percentiles are not a proxy for odds. And so what I have here on the left-hand side, we have a celiac disease relationship between odds and percentile. You see, if I came and said I was at the 80th percentile, my odds for developing celiac disease is 5. And this is largely driven by celiac disease polygenic risk being dominated by 1 or 2 variants, which with large effect. And this is what you see here, this sort of quick going up on the curve, right? Escalation of the curve. Whereas in prostate cancer, is sort of this incremental addition of variants, maybe towards the end of the curve, there's some variants of large effect included there. But if I was at the 80th percentile for prostate cancer, my odds are only like less than 2%. And so this is why we think it's really important that we communicate odds and prevalence as opposed to just saying someone's at the X%. Ancestry, you can't talk about polygenic risk without talking about ancestry. Or we know that polygenic risk models are Eurocentric. There's been some recent work that's shown like 60 or 70% of the models that have GWASs have been done Europeans. This is unfortunate. We made the following decisions on this: is we're explicitly, because we're running this as an LDT, we're explicitly calling out the ancestry of the discovery cohorts. We're independently validating only on European ancestry at this point, and all of our percentiles are using a European background population. You could ask the question of, well, why don't you go and develop new models using UK Biobank? Well, the answer is here on the bottom right. The bottom right of this slide is that the next most frequent ancestry, Central South Asian, only has just under 11,000 or under 12,000 samples. And that's before we even start filtering for deceases. So that really isn't big enough to be able to develop independently develop polygenic risk scores for them. How do we report this to clients? So on the left-hand side here, we have some verbiage around like what polygenic risk is and what health predisposition is. We make the point that this is not something that you change your clinical care based on, but it's something you consult your physician. You consult with your physician on lifestyle optimization. And so when we look at these 30 polygenic risk models, depending on whether they're female, male, 30 or 28, we basically break it up into those for which the client is greater than 90th percentile, and those which they're below the 90th percentile. And for the ones above the 90th percentile, we provide a lot more information, basically how your results compare to others. That's the percentile part of it, like what's your relative risk, and that's the odds part of it, like what's the heritability of the disease, right? And then finally, what's the lifetime risk, and this is kind of the prevalence part of it. And so using these four pieces, a physician will coach a client through their 90th percentile basal cell carcinoma result in this case. The way that this would be used by the clinic is maybe an increased screening, and maybe you need to go for dermatology screens more frequently, not every year, maybe go every 6 months. And the same goes for any of the other cancers. Maybe there's, in this case, it wouldn't be appropriate, but maybe there's a need to send out for Grail's test on a more frequent basis. And so that's the thinking of how this is used in a prognostic fashion and not necessarily a diagnostic fashion. Some of our findings, I promise this is the last pie chart that I'll show you today. On the left-hand side here, if you consider a finding or as anything where a client has a polygenic risk model above the 90th percentile, and we count how many clients across all of the ones that we've delivered so far have a finding in each one of the disease areas, we would expect them to all be relatively equal. And so this is what this is showing on the left-hand side. There are some obvious ones here that are not, and so at 11 o'clock on that color wheel is prostate cancer. Of course, it's only being issued for males. At about 2 o'clock is breast cancer, only being issued for females. And about 4 o'clock is endometriosis, also only being issued for females. The other aggregation we can do here is we can look at the number of findings per client. So are there any clients that are enriched for findings, and is that a deviation from expected? And so on the top of the right-hand side figure, this is basically the result of flipping a coin 30 times for every single client and asking a very biased coin and asking, are you in that 10% that has greater than 90th percentile? And at the bottom is our observation. And these two are in agreement. And so we're comfortable with the fact that they are in agreement. The last section that we have here is a nutrition and wellness trait section. I'll be fairly brief on this. This doesn't really need any independent validation because it's just straightforward extraction of an SNV on an indel. And what we're reporting out here to clients are traits that might be used in the conversations that they have with their physicians about vitamin statuses or vitamin metabolisms or food reactions or their approach to fitness and exercise, longevity and aging, and even disease susceptibility and the recent COVID-19 results. And so really, this is just a section to have some recreational content, but not to go as far as, are my earlobes detached or not? It does cilantro taste soapy to me. So we're kind of restrictive on the nutrition and wellness traits that we are including. That really covers like the content on how we've developed that content over the last 5 years. It's a lot of work stacked into like a 30-minute presentation there, but really what I hope to have achieved here is our thinking along, or how we're thinking with each one of these applications or each one of these report sections. I want to spend the last part of this talk going over our technology stack, and in particular the decisions that we made to move on to DNAnexus and how we made some of those decisions. So I'll start with what our current state is. And so at HLI, we built everything in-house, and fair enough, this was done several years ago. A lot of these sort of functionalities weren't available, and so we had to build these things from scratch. And so what we built out here is our own workflow systems. We ended up with 2 workflow systems, one for secondary analysis and one for tertiary interpretation work that was built on Docker. We were responsible, basically for maintaining our workflows, maintaining our infrastructure. We had to worry about things like, did that instance launch? Did we get a good spot price on that instance? Are we losing that instance because we lost the spot price? Did the run fail? All these sorts of workflow management and maintenance tasks we did internally. And of course, we also had to develop our own privacy and security program. I think that's irrespective, but we actually had to put in the code for access control ourselves, right? And then storage cost optimization as well. We had to do our own storage cost optimization, and then finally we had this across multiple environments. So we had a dev, test, stage, and prod environment where we were following a typical SDLC, software development lifecycle. And so the result of that is just for the clinical part of this workflow. Really what we had is like one SQL server, like 2 Postgres servers, like multiple DynamoDB tables, 4 APIs and many Lambdas that were all being internally developed and maintained. And so on the right-hand side here is basically the basic flow here. I'm just highlighting with what the asterisk is there is like, it's a database with an internal API, and that internal API has been developed by the team, and has to be maintained by the team. When we started transitioning to DNAnexus, and then this is where we landed on Nexus, the great thing about this for us is that we have one SQL database, we have one API that we don't have to develop. That's developed and maintained by DNAnexus. We don't have to worry about any infrastructure management. The only thing that people on our team have to worry about is building and deploying bioinformatics tools that are used in our reporting workflow. And so we focused really on how do we build and deploy those, and less so on are we getting the best spot price that we can possibly get for that resource on AWS, or are we making the best use of batch or any other AWS resources? We leave all those concerns over to DNAnexus folk, and they manage that for us, and we focus entirely on developing this part of the workflow. It's like, can we extract enough? What more information can we extract from a client's genome in order to add value to them? And so DNAnexus really allows us to make that transition. I want to geek out a little bit on how we do the developing part and how we develop with DNAnexus. So when we started looking at this, we started with a typical dev test stage prod environment, multiple environments, controlled deployment between environments, limited access on certain environments for developers. And so we wanted to mimic something like that, but without maintaining that complexity. And so what we did here is we captured a traditional software development lifecycle in one environment, basically. And so that's what's shown you on the right. I'd go through it very quickly. For those of you familiar with the DNAnexus platform, basically you have applets and apps. Applets are sort of more like development artifacts, where apps are deployable artifacts. And then on the workflow side, you have workflows, global workflows, and published workflows. And again, workflows are more the sort of development. They're combinations of applets. Multiple applets work together to create a workflow. But global workflows are combinations of apps. And then a published workflow is an immutable global workflow. And so we worked out this strategy using a combination of Git and testing both locally and on DNAnexus's platform that's shown on the right here. The only thing I really want to highlight on this is that in order for us to deploy an app, we have to go through a smoke test on DNAnexus, unit and integration tests on local, and some development that's captured on GitHub and captured in pull requests. And the same goes for a workflow. In order for a workflow to be deployed to something that gets used in production, if you like, it has to go through multiple tests, smoke tests, and stress tests, and all of that development is tracked on GitHub. So some of the other things that we do here is we ensure that we have semantic versioning on all our artifacts. The nice thing that we think we did here is we name all our applets by tool and git commit, as in this example on the left below. And what that allows is for multiple developers to be developing on the same tool and not overwriting their efforts on the DNAnexus platform. The other thing we do is we tag all of our artifacts on the platform with GitHub commit IDs. And what that allows us to do is say, well, here I have on the right-hand side, I have, for example, my run that I just ran on DNAnexus. I can see that it's tagged with a Git commit ID. And on the left-hand side, I can have that Git code open on that particular branch on that commit, and I can see exactly what the code's doing without having to go in and unpack the tool to understand it. And so really what we came up with here is a sort of controlled workflow deployment on the platform that mimics what we had before. The next section that I just want to cover briefly is compliance, and what made us feel really comfortable about the platform from a compliance point of view. Really just putting out two of the main requirements for us out of CAP, out of the CAP checklist for bioinformatics. The one is like patient confidentiality, security, and data integrity. And DNAnexus more than meets these requirements for us. Things like data is encrypted at rest and transfer. We have audit logs on everything. That meets a number of standards for data security and privacy. And there's a great white paper that you can read on the platform for that. The other thing is version traceability, and so we also had put a lot of work into tracing which particular version of a pipeline generated a result. The really nice thing about the way they've done this on Nexus is they've created the data objects themselves have full provenance. So you can query if I have a BAM file on the platform, I can ask you what pipeline generated that BAM file. And so this is really good for us to meet this CAP requirements. And so in general, we're really comfortable with both the security and with the traceability components of Nexus. I want to talk a little bit through how we did, or how we made a cost optimization decision on DNAnexus. And so this is really the result of a back of the envelope calculation that we did. We made some assumptions. So we looked at our current architecture, and we saw that our current architecture, in order to maintain it, we probably need to maintain and continue to develop it. We need two mid-range engineer full FTEs. And then we incorporated the architecture costs itself, and of course the variable costs with the sample. And so that represents, if you look on the right-hand side here, the very top of the blue curve, like 100% cost here is basically if we're running on our own platform at 10% of our current volumes, right? And so if we just transitioned onto DNAnexus, take into account the platform fee and also the cost per sample using the workflows that we have there right now, we saw 62% reduction in costs. And yes, that is the majority of that is made up by what is a platform fee versus what is the fee needed to have engineers maintaining your own internal platform. Of course, as you move right on both of those curves, they're going to decrease rather quickly, right? As you're hitting more volumes, your cost per sample become less, right? And so, for our projected costs at 100% of our current volume, we're looking about 68% more cost effective, and all the way up to like 400% of our current volume, we're looking at this being more or less 80% more cost effective. And the reason it becomes more cost effective over time is that we have put a bit of effort into optimizing the tools that we're running. The switch to Sention has helped tremendously. Sention's done a great job on optimizing both the variant calling component of what they do, but also the collection of QC metrics, basically using multiple threads instead of the single-threaded approaches of other tools. And really, just to summarize our migration here, right, what it allowed us to do is simplify our tech stack. Allows us to focus on product development, as I said before, right? We don't need to worry about maintaining infrastructure. We can just develop tools. It reduces our per sample and total costs. I showed that on the previous slide. And I think having an externally verified data security and privacy, especially when running an LDT and a clinical lab, providing support to clients like Health Nucleus, it's really important that we have this. I want to talk a little bit about what our future looks like. And so this slide, so far we've demonstrated value in HLI's Health Nucleus. We have piloted the genomics reporting with a few other clinics in Florida and Los Angeles. And so what we're looking at is to take this further than just the Health Nucleus. And so really what we want to be is we want to be an interface between a clinical care provider in the genomic space and genomic interpretation. And so I've spoken a lot today about our whole genome product. The important thing about that to notice is that, yes, there's a limited scope there. There's a limited scope based on our content in each one of those sections. But then we have this other product that's in beta mode at the moment that Health Nucleus uses extensively in their delivery for clients, and that's a genome search engine. And so this genome search engine, we upload all of the genomes into it, and it gives the ability for an end user to query a genome using a very simple syntax and a Google-style interface. And so this is not something that I could demo today, but certainly something that we can look at in the future. And so what we're thinking is this combination of products, the one with limited scope that's more clinical in nature, and the unlimited scope that's more discovery in nature. This can be combined in providing care to clients that are being seen by physicians in the clinic. And so we're looking for the potential to expand this. And so for anyone that's interested in building this sort of model, just contact me on LinkedIn if there's any further interest. And really that's everything. I'm just about getting to time. Yeah, that's everything we want to talk about today. I've had the phenomenal opportunity of working at HLI for the last 6.5 years, and associated with that, I worked with so many talented and exceptional people that have been through that company. And so I just want to shout out, and this is just a shout out. I've probably left some of them off here, but this is just a shout out to all the people who've had some hand in some component of the product development that I've spoken about today. Of course, I just shout out to DNAnexus as well, and to GenomeWeb. Thank you for this opportunity, and thank you for a great partnership that we have with you. And that would be it.
Great. Thank you very much, Dr. Delport. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to take a moment after the webinar has ended to give us feedback by taking our exit survey. All right, let's jump into our questions. So Wayne, how have and how will sequencing technologies, particularly long-read technology, and also automation, affect your efforts and your success, do you think? And will the overall cost of sequencing need to continue to drop for you to continue to be successful?
That's a great question. So I'll tackle that. The first part of it is the long read combined with short read technologies, right? I think the future is combination of those. I think for long read technologies, yes, we're going to have the ability to do genome assembly for every single client. Is the reference genome caught up with that yet? No, it's not, right? We still have a single reference genome. And so this is, and because we still have a single reference genome, all the information out there about variants is captured on that reference genome. So I think that process is going to be rather slow, because we have to then translate variant information on a reference genome to this newly established genome for a client by doing an assembly. But I do think that is the way of the future, especially when we start looking at larger structural variants that are very difficult to call with short reads. There are some other great platforms that can do structural variants. We could talk about that for a long time, but that's my thinking on how long reads impacts this. Does our cost per sequencing, does it need to go down in order for us to be successful? We're making a bet that the cost of sequencing is going down. We've seen dramatic reductions in cost of sequencing already. I think the Illumina's $1,000 genome this year. We know that sequencing is going to get cheaper, right? There's no doubt about that, and that's the bet that we're making. We're focusing our efforts more on the interpretation side of things, so extracting the value from the genome, than on the more commoditized things such as doing the sequencing itself or doing alignment and variant.
So what's been your experience with data reanalysis in order to account for new information or updated literature, in particular to account for the reanalysis of variants of unknown significance? Do you have like a predefined frequency for reanalysis, an annual regeneration of reports to include new variants, anything like that?
Yeah, another great question. So yes, we do have a policy for reanalysis of the USs. I would be lying to you if I could tell you exactly what that was. It was either 6 months or a year. It's in our policy. So we will do a reassessment on VUSs that we've identified before in that time period. In terms of reanalysis for reporting purposes, the model that Health Nucleus has built out is a continuous engagement model, which means that a client comes in for base testing, and then over time will come back frequently, whether that's annually or every 6 months, for another evaluation. And as part of that evaluation, they would be able to order an updated genome result. Of course, we've sequenced their genome already, so we don't need to do a resequence. And so then it's just a matter of re-annotation and reissuing the newer version of that report. There is, using a static variant list like we're using, there is a potential criticism here, and like, well, what about variants that are unique to clients? That wouldn't be reported immediately to the clients, and this is why we provision this additional tool for the clinic to be able to go and review the client's genome. But more importantly, if they do find anything, we give them the ability to report out on those findings through a single variant report that we still run under the LDT. And that's another piece that we didn't cover today. So we're working towards having this more streamlined. But at this point in time, the only way you'd get a reannotation or a regeneration report is if a physician reordered.
Have you been able to qualify or quantify at all the benefit of genomic diagnostic reports using whole genome sequencing as compared to whole exome sequencing, maybe by defining a diagnostic versus prognostic value index?
Yeah, we have not done that. I mean, that is certainly something that we need to do. Just thinking off the top of my head, our variant content, our hereditary variant content, would probably be similarly achieved with an exome product. Maybe some of the polygenic risk models are going to be a little bit more difficult to do with an exome product, given that a lot of the variants are outside of the exome there, because it's more about what those variants are linked to than the actual disease-causing state of the variants themselves. I think for our pharmacogenomics component, and this is why we initially steered away from exome, is that the capture process actually does influence the copy number estimation for CYP2D6. And so that's rather difficult to manage, and we didn't optimize that. So we're continuing with whole genome at this stage because we want to do more than just what we're reporting at the moment. There's short tandem repeats that we want to get into, and a number of other applications that wouldn't be suddenly covered by exome.
Could you walk us through how samples are collected, if there's any sample level quality control that's done before the assay is run? And in particular, since you're using PCR-free methods, how much DNA is required for the assay?
Oh man, that's a question for our lab. So samples are collected in health nucleus, right? Of course, we collect whole blood, and single valve whole blood is submitted to the lab. Those have dual identifiers, basically two IDs in order to track the sample. An independent shipping manifest is sent along with the sample, as well as an electronic manifest is sent to our lab, and so that allows us to check up that the samples that the lab received as the samples that was intended to send. From the point of view of the exact amount needed, I might have to get back to you on that question. That's a wet lab question. I should have that at the top of my head, but I don't.
No problem. Thanks. So that's fine. Are you able to tell us how access to biosamples and data in the sequencing lab is limited in order to preserve privacy and safety?
Yeah, so everything is completely de-identified. There's no association between a client's ID and client sequencing ID and the client's name and PHI. All of this is done in an access-controlled production environment, limited access. The only people that have access to these data are people that are operational in the clinical lab. In addition to that, any R&D work that we do on those samples, and by R&D, I mean product R&D, we do on a completely de-identified data set where the link between the randomized ID is completely broken from the link of the sequencing ID. And the reason we do that is because we don't want to have to worry about that R&D data being informative of any of the clients in the lab or in the clinic.
And then we have a question about the cost of whole genome sequencing, including the complete report. Is that something that you can tell us?
That's not something I can share right in this forum. The costs we're running, I can give a broad range. I mean, more expensive than your typical exome, less expensive than a diagnostic where there's manual interpretation. If they know what those are, yeah.
Fair enough. So have you tried any calibration approaches for ancestry adjustments of polygenic risk scores?
Yeah, it's a great question. So 23andMe did something like this with their type 2 diabetes model. There's been some recent work, I think last week, that was published in this space, and so we haven't formally explored this. We have looked at the idea in principle. I think we're still at a case where we're limited by the appropriate data availability. Certainly for some of the more common diseases, it might be possible using UK Biobank and using some of the more common ancestries or more common non-European ancestries there. But really, I don't think we're there in terms of the data yet. I think we just have to put more effort into finding appropriate data sets and registering for access to more biobanks. With that in mind, there is this recent paper that came out last week, I think came out of University of Michigan, And really the take home from that paper is yes, the absolute PRS scores do change, but the risk stratification doesn't. So you're for a European model that's been applied to a Southeast Asian or a Central South Asian population, the PRS distribution will shift, but the ranking within that distribution stays the same. And so that is fairly encouraging.
And then what's next on your product roadmap? Can you walk us through what features or content you think aren't adequately represented at the moment that you think are coming up?
I mean, you already touched on some of that. I think the ancestry-specific polygenic risk models, have an obligation to make that better for non-European clients. I think that's forefront. The other thing that I didn't really I touched on, but I didn't really go into details on, is adjacent variants where the interpretation differs depending on whether those variants occur in cis or in trans. We think about these as variant couples. We need to make the reporting for that automatic as well. So those are the two things that we're thinking.
Great, thank you. And we have reached the top of the hour, so I'm going to wrap it up there. We'd like to thank Wayne Delport and our sponsor, DNAnexus. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.